Have you ever stopped to think about how we actually make sense of our DNA? You know, that incredibly complex code that's packed inside every single one of our cells? Well, today we're going to pull back the curtain and dive into exactly how computers manage to read your genetic code. So, to really get a handle on the challenge here, let's just start with the sheer scale of it all. Your personal genetic code is like a book written with 3 billion letters. Nope, that is not a typo. 3 billion letters. That's the amount of data that makes you, well, you. It's just a staggering amount of information. Okay, so if your genome is this 3 billion letter book, how in the world do scientists find a single tiny typo? A one-letter mistake that could be the absolute key to understanding a disease. That right there is the central problem we're trying to solve. And the answer, the field that makes this all possible, is called bioinformatics. This is where biology, computer science, and statistics all crash together. It's the powerful science that gives us the tools to actually read that gigantic genetic book and hunt for those meaningful insights. So, where does it all begin? Let's trace the journey of your DNA, right from a biological sample all the way to becoming digital data that a computer can actually understand and analyze. It really boils down to a 3-step pipeline. First, a sample gets collected, think saliva or blood, and the DNA is pulled out. Second, these huge sequencing machines read the DNA, basically turning the physical stuff into raw digital files. And finally, that raw data gets a good cleanup and is aligned against a standard reference genome so we can start to see what makes it unique. And as you can probably guess, this isn't happening on your everyday laptop. Oh no. Scientists are using a massive digital toolkit. We're talking powerful supercomputers, specialized software, and coding languages like Python and R just to manage and really dig into this data. It is a seriously heavy-duty operation. Okay, so now we have the data. This is where the real detective work begins. Let's look at how computers actually sift through billions and millions of letters to find the few that really matter. Now, the official term for one of these typos is a genetic variant. It's just any spot where your DNA sequence is different from that standard reference we talked about. And these variants are exactly what make each and every one of us genetically unique. Now, this is where things get really, really cool. The traditional method, GATK, it's kind of like a spell checker that follows a very specific set of rules. But the new AI-powered approach, DeepVariant, does something amazing. It literally turns the DNA data into an image and then uses powerful image recognition to visually spot the typos. It's like facial recognition, but for your genes. And this makes it unbelievably accurate, especially in the really tricky, repetitive parts of our DNA. All right, but here's a twist. Not all typos are created equal, and finding them requires 2 totally different detective strategies, depending on whether that typo is super common or incredibly rare. And the key concept that splits these 2 paths is something called allele frequency. The best way to think about it is like this. If everyone's DNA is a book, is this typo a common misspelling that you'd find in half the copies out there? Or is it a unique 1 in a million printing error? That distinction changes absolutely everything. So for those common typos, the ones that are like those widespread misspellings, the go-to strategy for scientists is a method called GWS. A GWAS is a massive, massive study where scientists scan the entire genomes of thousands, sometimes even millions of people. They're basically playing a giant game of spot the difference. They're looking for common variants that show up way more often in people with a certain disease compared to people who don't have it. But for those 1 in a million rare variants, the approach has to be much more targeted. Instead of looking at millions of people, scientists might trace a single variant through a family tree. Or they might group a bunch of different rare variants in the same gene to see if their combined effect is causing a problem. They also rely on huge databases where experts have been cataloging these rare finds for years. So, we've gone through all this work to find the variants. What's the payoff? Let's bring this all home and look at a powerful, real-world application that shows what this all means for your personal health. It's called a polygenic risk score, or PRS for short, and it's exactly what it sounds like, a single number that sums up your genetic likelihood for a specific condition, like heart disease or diabetes, based on thousands of those common variants. Now, this is the crucial part. This score isn't just pulled out of thin air. It is built directly from the results of those massive GWAS studies we were just talking about. It uses the data from all those thousands of people to figure out what your personal risk looks like. And the way it's calculated is actually surprisingly simple at its core. Each one of your variants is given a weight based on how strongly it's linked to a disease. Some add a little bit of risk, some are protective and actually subtract a little risk. You just add up the effects of all your relevant variants, and you get a single, personalized risk score. And really, this is the ultimate goal, the big idea. The whole purpose of these scores is to help flip the script on medicine, to move from being reactive, treating diseases after they happen, to being proactive. It's all about personalizing prevention, using your unique genetic code to help keep you healthier for longer. And this really brings us to the cutting edge of modern medicine. We are getting better and better at predicting our genetic future with some real accuracy. And the question that leaves us all with is that a good thing?